Record button. Yeah, so this project is essentially a discrete diffusion method, and we have tried to use it mostly in the context of biological sequence design. And this project is also kind of adopted from the discrete walk jump sampling paper by the GeneTech company that was published in iClear, last year's iClear. And I will start with like the motivation, then I'll go to some preliminary ideas, and then I will describe the method and some of the results. And feel free to interrupt me anytime if you want, if you have any questions. Also, you can use the chat box to ask questions. And this was published in TMLR, and before a preliminary version was presented in iClear workshop. Okay, so why do we need antibodies? The reason that I'm asking this question is because the main idea from this project came from the discrete walk jump sampling paper, and they mostly used antibodies. So we will start with that, but it could basically be generalized to any biological sequence tasks. So the reason why we need to generate antibodies is because we need to get antibody-based drugs, and there are multiple considerations. This is important because it will be important in the later part of the talk. Here we need the efficiency, we need the safety, and we need the manufacturability. That means the antibody that we actually need to be useful in silico need also be synthesizable so that we can use it in vitro. And the problem with antibody generation in general, or the protein sequence generation, is that there are many multiple variable regions. So there are some regions, some parts of the antibody, also in some cases nanobodies, that where you have some regions where like a change of a letter might be a lot of change. So, and then another problem is obviously their experimental validation is really time consuming. I checked synthesizing antibodies, and like one of the antibodies will like it takes almost $1,000 to just check one antibody. So it's really expensive. And it also takes a lot of time. Like it at least takes 2 months to synthesize one antibody inside a rat. So what we could do is instead produce a lot of antibodies in silico, validate them, take the best of them, and then try to synthesize them and see how many we can actually get. And that's the basic idea of all the generative model-based protein sequence design in general. So the other thing that we have to consider is whether we want to generate antibodies in the structure space or the sequence space. So the good thing about structure, obviously, is that you have a structure and it is continuous. So it's in the continuous space. That means that you can directly optimize it for a specific antigen. So the problem with that is that if you have the structure, you will still need to convert it back to sequence. That's called inverse folding. And the problem with that is sometimes the structure doesn't guarantee a sequence. You might have a structure that seems legit, but when you try to convert it back to sequence, it doesn't work. And even if the sequence exists, the generative model that we have nowadays doesn't have the best inverse folding capabilities. So it could still fail. So the other way would be simply to optimize it in the sequence space. The problem with that, obviously, is that sequence is discrete. And that's a problem, obviously. So diffusion will have some issues with that because diffusion is mostly built on the continuous space. And the other problem with diffusion is that, you have this noise scheduling that happens in the diffusion model. And the autoregressive model has this next token prediction. That means you will accumulate errors as you go. So there are these two problems with two different methods. So what could we do? Well, what if we could have a diffusion model, but with no scheduling? So that's what we're going to focus on here. Some basic stuff. So this is the delta nabla log p, which is the direction. That's a gradient of reducing noise. So that's a denoising gradient. And to learn the score. That's also called the score, which to learn the score, we essentially add some noise to the original data and learn the perturbation. So basically we learn how to get back the original data. That way we train the model. That's score-based model. That works well for the continuous data. That means this y here should be continuous. The other thing that we can consider here is that neural empirical Bayes, which essentially is a way to add noise in a way that is recoverable. So that means this x here that you can see is discrete. And then we add some noise to it. There's a Gaussian noise. That means when you add that noise, the y is actually continuous. And you can see there's a formalism, which is the approximation of x given y using some formalism, but that formalism should look very similar. That's again the same nabla log p, but this part is actually the score model. So that means if you could learn the score model, then you can have this noise y and you can get back the x. That means you can essentially get discrete data jumping back from the continuous data. So that's really the idea of discrete walk jump sampling. You have the discrete data that you get from your data set. You add some noise, you get continuous data. Then you can do this, denoising walk of the basic diffusion. You have this MC walk on the continuous noisy data. And then when you're done with the walk, you are still at the continuous space. So you still have to jump back to the discrete space. And you do that jump using NEB, neural empirical Bayes. The good thing about this is that you can essentially jump back anytime. This is just not something that you could do with diffusion, for example, because like diffusion has a noise scheduling. That means you have to finish the schedule to actually go to a data. Here, you can keep walking as long as you want, and then it's up to you when you want to jump. Now, so the idea that I presented before.
So sorry, Zarif, what's the full name of NEB again?
Neural empirical Bayes.
Okay, get it.
Okay, great. Any other questions? Okay. So the idea that I presented before is great for generating pool of data. If you have a big data set, the problem with that is that the things that we consider for biological sequence design are not usually just mimicking the data present in the data set. It's also usually to optimize something. That could be the efficacy, that could be some sort of manufacturability parameter, that could be a specific binding affinity to some antigen. So we still want to have that optimizability of our method. And that's really what this method does that I'm going to present now. So it's going to look really similar to gradient guided diffusion, because mainly that's where the idea came from. And then we add some formalism to adopt to the jump part, because that is obviously not obvious how to do. So as I said, our generated sequence need to be antibody-like, but also need to be optimized for a specific attribute. And how can we learn attributes? Well, we can have a discriminator model that could take care of the attributes. That means you pass in some data, and then your discriminator model tells you what the attributes are. The interesting thing here is that you can see that f theta takes y. And as we learned before, this y is not the data itself. X was the data because x is the discrete variable. The y here is continuous. That means this y is going to be the noised version of the discrete variable. That could be useful later. Okay, the attributes could be anything really, from antibody affinity, aromaticity, instability. In some of the experiments, we also use attributes as in a proxy for how the model binds. And then if we have a model that's differentiable, then obviously we could take the gradient of that model to go towards a attribute so that we can maximize it. That's a gradient guidance. So here I have tried to color code it. So usually the discriminator model is going to be red and the score model is going to be blue. So idea is very simple. We learn a noisy discriminator model. As I said, it has to be noisy. That's like a subtle variation from the diffusion model. Then you have to learn a noisy score model. And then you just add the gradient guided walk and gradient guided jump. Now I'm going to describe how these things work. So first we have to learn the noisy discriminator model. You add the noise to the discrete sequences. That is, you get some data. Let's say you have a 20 length antibody sequence, and then you will have some sort of one hot encoding of that. And if you have the one hot encoding, you can simply just add some Gaussian noise to it. That means now it's not going to be a one hot encoding. It's going to be some continuous representation of that. And then if you have that continuous representation of the entire data set, you're going to take that data set and learn the attribute that corresponds to the discrete data. And then you learn the predictor on top of it. Next.
I have this question. Why the discriminator is on Y, not X directly?
Yeah. The reason is that when you're going to do the gradient guidance, so think about when you actually have to do the gradient guidance. It's when you have to walk. That's an MC-MC walk. And then MC-MC walk actually is defined on the continuous representation, not the discrete.
Yeah, I know.
And so if you have the MC-MC walk and you use the gradient, then your gradient should also be defined on the continuous space. That's why we need to train the discriminator model on that.
Yes, I get it. So it's like your of course the discriminator is really on the X, but X come from a jumping from the Y space, noisy space. Therefore, on guided MC-MC, you want to make the walk towarding a higher score. I'm sorry, wrong. Okay, higher reward for your discriminator, and therefore it's easier to put the discriminator in the Y space. That's the reason, right? But I mean, my point is, but it's actually the thing that give you a reward is really your X space stuff.
That's correct.
So I'm just curious if this if we do a version of discriminator on X versus a version of discriminator on Y, what would be, is there a big gap in sense of performance? For example, if you do a discriminator on X and somehow link that reward to the random walk noisy space in Y space, is that better than directly doing on Y space? So that's my question.
Yeah, I think I do get the question. So I have thought about that. I haven't tried that, but I do believe that it could be useful. What I have tried though is just simply trying to learn a discriminator on the X space, and yeah, that didn't work. So it still needs to be defined on the Y space, but yes, the way that you define it can change. For example, what you said could be also a valid solution.
Continue.
And Srinivas asked a question, is a gradient guidance similar to classifier guidance? Yes, it is really similar. The two differences that I can think of is that one, classifier guidance usually will be defined on the same space as the data. And the second is that the classifier guidance doesn't have a jump part. So we have some subtlety to the jump part, and we have performed a mathematical validation to it as well in our paper. So we had to formulate the jump part with gradient guidance as well. So we have a gradient guided walk, which is similar to the classifier guidance, but we do also have a gradient guided jump to go back to the discrete space. So that's the difference. Let me know.
Thank you.
Okay.
But I have one doubt. The issue with classifier guidance is that it generates high quality samples, but they're kind of all similar.
Exactly.
Like
Yeah.
it doesn't cover the mode. It
Mhm.
doesn't, it's a problem in this application.
Indeed, it is still a problem. So in our paper, we do have a lengthy discussion on that. So
Okay.
there's a
Sure.
key thing, really. There's fidelity, and then
Mhm.
there is optimization, that's the quality, and there is the uniqueness. So these are the
Okay.
key attributes, I think, for generative models that are, in tension.
Yeah.
So for our work, I think we covered the fidelity and we covered the quality. But again, if we want to handle these two well, then the uniqueness suffers. But if you want to handle uniqueness, then your quality will suffer. So there's this tension.
And one more thing, in the like 2 slides before, while introducing this class gradient guidance, you you discuss some motivation. I didn't get that motivation, like why you went for this gradient guidance. Can you please repeat that if you don't mind?
Sure. Yeah, so the real motivation is, okay, I'll start with the motivation of the previous work that we based it on. So the motivation for that previous work was that even if we have a big data set of antibody sequences,
Mm-hmm.
if you just perform a simple discrete diffusion on top of it,
Mm-hmm.
what happens is the generated samples, especially they show that the generated samples suffer from antibody-like properties. And so what they want to do is they want to tackle the fidelity challenge. That is that the
Okay.
generated samples should be similar to antibodies. The problem
Okay.
with that approach is that we also want sequences that are antibody-like, but also they
Mm-hmm.
have specific qualities. So
Okay. So it's mainly like improving the quality of the
exactly.
data sample,
Mhm.
more like the training data, right?
It has to be more like the training data, but also it
Yeah,
has
but
to,
something different.
uh, has to have some specific qualities. Yeah.
Okay.
Mhm.
Okay. Thank you.
All right. Anybody? Okay. All right. I am going to go forward. So yeah, we were learning a predictor on the noise data, and as Dianbo mentioned, that yes, it is also possible to learn the predictor on the X, that is the discrete data, which is the true label, but also connect it to Y with formulation. I can think of the Gaussian formulation as I speak, but yeah, it could definitely be done. And that could also be a nice avenue for future work. So next is again very similar to the previous work. We simply add noise to the discrete sequences and we train the score-based model, but on the noise sequence so that we can walk using MCMC. And also, I should mention this. This is specifically Langevin-based discrete MCMC. So there's like a little subtlety between the discrete MCMC and the original MCMC, but yeah, that's really subtle. Next is the gradient guided walk. It's going to be similar to the classifier guidance. So you simply have the MCMC to denoise, but as you denoise, you can have your score-based gradient. So that means you pass the noised version. So you start with a very random sequence, basically a Gaussian sample. And then from that, you pass it to the denoising model. That denoising model is going to give you some gradient. And you will pass this Y to your discriminator as well and take the gradient as well. So your torch autograd is going to take care of that. And if you combine these two gradients, that's going to give you a direction that should help you to go towards the denoised version of that, as well as it should also lead you to some optimized version of that. And yeah, so we walk in the smooth manifold, which is to say that we are in the continuous space. And so the score-based model should work as well as the discriminator. And there is the gradient guided jump. So you have the NEB, I have mentioned that. We also formulate a conditional NEB, which is essentially a way to use a gradient guidance as well as a score-based model to perform this and perform this neural empirical base, but it's a conditional NEB. So you can, we have the proof in our paper, but it's essentially just you add the gradient, discriminator gradient for the guided jump. So that's really the full picture of our method. So you have the small dots there on the left. That's the discrete space. you add some noise to go back to continuous space. As you can see, shaded colors. The red colors, pinkish colors, mean that we want to be in those space because that are more antibody-like. So we have two of our gradients. The pink gradient is taking us to the pink zone. That's the hot zone. And then we will have this gradient guided walk. And then finally, with the gradient guided jump, we will jump back to hopefully the yellow dots, because those are the bigger dots. And those are where we want to be. And with different gradient, that is with different quality of the attributes, you can either be on the left-hand side of the pink zone or the right-hand side of the pink zone. So that's our gradient, discriminator gradient will do. So we also try to use this in another case. So far I was talking about optimization. When I was talking about optimization, it was mostly about single objective optimization. So we had just had one objective, but we could easily scale it up to multiple objectives as well. So the multi-objective optimization would simply be to have D different rewards. You, instead of having just one reward, you just have different rewards, and you're trying to maximize them all at the same time. And that's what I just wrote here in a mathematical form. So what could we do about that? So we could take the multi-objective optimization and then decompose it back to a single objective. So that means somehow combining them together so that you just have one reward. And one easy way to do it would be to just have a weighted sum of it, which is also known as, which is also known as the preference conditioning. And we can try to approximate it given the weight with a neural network. So this neural network is going to be conditioned to the weight, and that should give us proper output of the preference conditioned multi-objective. So how can we do that? Using our method. So you can see that we could essentially, if we had like multiple rewards, we could have like D different models predicting that. And then we could have like the gradient of all the D different methods. And we could take the gradient guidance of that and then use our model. Or we could sum them all up, since all of them are essentially working on the same Y, and then we can just approximate it with one model and then take the gradient of that given the weight. So those two different things should be the same mathematically. And that's really what we do here. So everything stays the same. We still have our gradient guidance. And for the gradient here, we learn a model that is preference conditioned. And then we take the gradient of that discriminator. So first so again, think about the data set that we have. Now think about, we have like D different attributes of that for specific antibody. And we combine the attributes with the weight. And what we do in our case is like we have some random weights to it, assigned to it, and then we make another data set, and then we learn the discriminator for that, given the weights. And then in the inference time, for multiple preference weight, you sample a lot of sequences, and then you create a Pareto front. A Pareto front essentially means that the samples that are the best among all the other samples. So yeah, okay. I think I have a question. W1, yes, okay. Yeah, sorry about that. So yeah, if we have like D different models, then we will have the weight for each of the attributes. That means even think about like if I have the binding affinity for 3 different antigens for one antibody, then the preference would be that which one do you prefer the most? So would you like the antibody to bind with the third one most or the first one most? So given that, that's our weight. And that is also called the preference. So this is how this works. And you can condition on the preference, and using the preference, your model is going to tell you which way to go. That's what we will try to do here. Does that answer the question? Srinivas? Okay.
Hello, Zarif. Can you hear me?
Yeah, I can.
Could you explain the background information of there's some echo. Could you explain the information about the what is walk and jump in this scenario? What's the walk and jump meaning?
Okay. So yeah, I think I get the question. So here, what we are trying to do is we're just approximating the gradient. So you can see that this gradient is still the same. This is the score-based model. And the right-hand side is the discriminator and the gradient of it. So if we have these two gradient, now you can think about the previous picture that we had in our mind. We were using the score-based model gradient and the discriminator model gradient for our MC walk. So that would be our gradient guided walk. And for the gradient guided jump, we were using, remember that we were still using the discriminator gradient there too. So that would go right there. Does that answer the question?
Is this used in the sampling process of the in diffusion model or autoregressive model?
I would assume that it's more similar to diffusion.
Okay.
Yes, in the sampling, yes. We're approximating gradient so that we can use it in the sampling. So inference time, yes.
Okay.
Does that answer the question? Okay, I will go forward. So I'll go with some results. Most of the results.
So I actually got a few questions before we move to the results. So,
Yeah.
just can you remind me the editing noise to discrete? Okay, the moving from X to Y step, is that a single step of editing noise or is multi-step?
It's a single step. Yeah.
Single step. Okay. So if I understand correctly, the overall picture is you move to X to Y by adding single step of Gaussian noise. Then there are two steps of denoising. One is gradient guided MCMC in the Y space, which gradually move to a low energy area, which near a real discrete space thing. And then you have a single step of jumping from discrete space to from continuous space to discrete space. So that's also, and let's go even narrow down to the exactly estimated X space. So there's two steps of denoising, right?
Yes.
And now you are only adding the guided gradient and the multi-task thing. So and I have three questions about this whole process. Number one, while there is a reason we are using multi-step noise for diffusion. Here is a single step adding noise is a problem. Number two is MCMC the best way to navigating in the Y noise continuous space in the denoising process. Number three is that single step of jumping, is that the best option? Or we want to do multi-step jumping, or it's a more complicated jumping? So that are my three questions.
Yeah. Okay. I will start with the first question. I think I already forgot. Could you please remind
So the first question is, you are only doing single step adding noise. Is
yeah.
that good or bad? Yeah.
Okay.
Well, it's multi-step.
So yeah, I think I get the question. So the main motivation of the base paper of the discrete walk jump sampling actually was that we don't want to do the noise scheduling of the diffusion. So we instead want to try out how the one step adding noise works. And I feel like with more noise scheduling, this method should improve. So this is really a spectrum, like how much you want to do the noise scheduling. For this one, this is like on the other end of the spectrum, that you just add one step noise and try to denoise it. And I feel like that if we add more noise, if we would add in more noise, I think it would help us more with the uniqueness of the model. But I would think that it might suffer from the fidelity of the model, because we're losing too much information with adding more noise.
The second question is, MCMC is using to navigate its noise space. Yeah.
Now this one, yes, this one actually is interesting, because obviously we could essentially use all the optimization technique that we have. So that means we could, MCMC could be one, but there is also, evolutionary strategies that could also work there. The only problem would, I think, in my opinion, would be that there are just too many parameters to optimize for that. But yeah, those would also precisely work. MCMC is just common for diffusion, but I also, again, as I just mentioned, I also saw a paper showing like evolutionary strategies working just as fine for diffusion too. So that could also work.
I think one problem of using MCMC and other sampling method is if you have multiple peak.
Yes.
Like the figure just showed, if both of the right-hand side and left-hand side red color peak, if they're both valid, then it is likely your model going to stuck in one of the peak.
Yes.
Yeah, okay.
I guess the good thing about the gradient guidance is that if it helps narrow down the directions to go, because like if you don't have a gradient guidance, you have just too many directions to go. And assuming that having a specific attribute narrows down the places to go, it should help us with that. And also we start from random places for our initialization. We start with Gaussian noise simply. So that could also potentially help us with the uniqueness problem.
Oh, okay, good. I mean, there might be multiple peak even after your guidance, right? So it's like, I think the MCMC still suffer from it, because when
yes, it would definitely suffer from it, yeah. I
think that's also a problem. So okay, the third question I asked, it was about you have the after MCMC sampling, the jumping back from Y to X is also single step, right?
I asked, yes, I think, yeah, I can answer this question. I had the same question too, and I asked the main author about this question in the iClear. So they said that they tried multiple jumping too. One, mathematically, it's not valid. So the author also authored the neural empirical base paper. So they said that mathematically it doesn't make sense. And second, they tried it, but yeah, there is some improvement, but the improvement just gradually diminishes as you increase the amount of jumps.
And maybe is that because the reason is mathematically invalid, is that because they only use a single step of adding noise? So
yeah,
the jumping back, of course.
Yes.
Okay, good.
The other thing that the author mentioned also that it could be interesting to try out jumping, but to try to jump to multiple places. So, the equation that we have is completely deterministic for our jump part, but you can make it a stochastic equation too. And then if you have a stochastic equation, then you can potentially jump to multiple places. And that could be interesting too.
So that will become an inference time compute jumping. And the more jump, the pick the best of n, then you can get the best result. Okay, get it. Yeah. I mean, the MCMC is also stochastic.
Yeah, that part is stochastic, but the discrete part is not. Continuous to discrete part is not stochastic. So that could also be made stochastic and see where we can jump.
Good. Okay, yeah. Please continue.
Yeah. Anybody has any other questions? I assume not. Yeah, for the results, I'm going to be really brief. We have the full set of results in our paper too, but I'll just go over some of the results here. The intuitive results, that probably is the discretized MNIST. So we were trying to first just start with some easy experiments to see if our method even works. And the discrete MNIST was the best, because it is difficult, because the original MNIST has the continuous information, but when you discretize it, so you basically have this threshold. After above 0.5 is 1 and below 0.5 is 0. So it's black and white. And you can see it on the left-hand side. On the left-hand side, you can see that that's the gradient guided, just the walk jump sampling method. On the right, we have the gradient guided walk jump sampling, and you can see that the top row is the 0, the middle row is 3, and the bottom row is 8. And you can see that it's not perfect. In the middle row, you can see some of the 3s have turned into 8, for example, and some of them has turned into 9. So it again shows that this method is not perfect. And this is also, this is something that we played with in our paper too, like how is the strength of the gradient? So if you increase the strength of the gradient, then the images are going to be more specific to the condition. But then again, the images are going to look very similar. So there's this tension that we have. Then we tried the exact same experiments that the walk jump sampling paper did. So you can see the discrete walk jump sampling paper, DCS. I probably should take a while to also explain what DCS is. DCS is similar to, it's going to be similar to FID for images. So it basically tells us that how close the antibody sequences are and how to the original data set, as well as how antibody-like the antibody generated sequences are. And we do it not by just looking at the sequences itself, but also the attributes of the sequences. So we basically, to calculate it, we have the algorithm in our paper, but to calculate it, essentially what you do is we take the reserved data set that we haven't used, and we find a distribution of some specific attributes, and we try to gauge how close are the attributes of our samples to that distribution compared to the training samples attributes. And so we have this comparison between these two, and we find out if they're better or not. And that's DCS. So we obviously want DCS to be as high as possible. That tells us like if our method is producing antibodies that are more similar to the, that are more antibody-like, essentially. So we want the DCS to be high, as well as we want some specific attributes to be optimized. In our case, the instability index was one of the attributes, and the percentage beta sheet was another attribute. One other note is that the percentage beta sheet might look like a attribute that has been calculated to the structure, but it really is not. This is more like a calculation based on some heuristics. So it's not calculated on the structure, just so you know. It's just on the sequences. You can easily calculate these attributes by the packages. I forgot the name of the package, but yeah, it's a very common package. Okay, so you can see some of the results. So we basically had the same baselines as they had in the paper. So they used VDM, and then they used the IGLM. IGLM is a language model on the antibody sequences. They also apparently use GPT. I really don't understand why they would do that, but they did it, so we also did, and we also tried gFlowNet to perform some optimization on the sequences. As you could see with our discriminator, specific discriminator will optimize specific attributes. So this is a single objective optimization, and you can see that in our both our discriminators, we actually get sequences that are more similar to the antibodies. And interestingly, with gFlowNet, we did get okay, so for gFlowNet, there is also a side note. The antibody sequences that we were dealing with were too big. So that was like 250 letters. So gFlowNet just couldn't handle that. So we still had to show it in our picture, but it really, yeah, it couldn't handle that. Too big for a scaler. We also tried the AMP task that probably will be more similar to the gFlowNet community. So this AMP task is presented in the biological sequence design with gFlowNet's paper. That's the first task. And so we picked the same results from that paper, and we also added our method to it. So yeah, AMP task, it's exactly the same experimental design as the GFlowNet task. So you have this AMPs that are training sequences, and you have another oracle that is trained on some other reserved sequences. And so we use the oracle as like a reward model here. So in our case, we don't use the oracle for gradient guidance. We have our training sequences and we learn another discriminator, and then we use that discriminator instead for our gradient guidance. And so we use that, and we basically see how the performance, diversity, and the novelty is. And this is also a good thing to show the tension between different attributes. So you can see that the performance and diversities are really good, but the novelty suffers. So yeah, that is something that we notice throughout our old experiments. And but again, the performance were pretty good, and you can see that the known AMPs were more or less close to the generated AMPs. That is really important for biological sequence designs. So you can see the distributions were almost matching for both of the cases. And we did perform a lot of other multi-objective optimization experiments, but I'm just showing one here that you can essentially, as I told you, have sample different preferences, and for each preferences you sample a lot of antibodies. So you can see the blue ones are all the samples, including the red ones actually, but the red ones are the ones that are covering the region. So these are the Pareto fronts, and so you can have these Pareto fronts with our model too. In our case, the two attributes that we picked were instability index and beta sheet percentage, but in our paper, we also picked like 5 attributes to scale it up and see if that works, and it works. So yeah, you can find everything else in our project URL, and we can also find this slide, by the way, too, there. And yeah, with that, I think I can conclude my talk. Thank you to all for listening to the talk. And if you have any other questions, and if you have any other ideas and you want to collaborate or extend this project, feel free to let me know. Or Dianbo.
Thank you so much. It's a very nice talk. Anyone have any other research questions for Zarif?
Zarif, you already explained, but I didn't get
Mm-hmm.
what's the difference between gradient guidance and the existing classifier guidance?
Mm-hmm.
Sorry, what's the difference? I didn't get, sorry.
So yeah, the two difference really is that the first, if I have to explain this through classifier guidance, how can you change the classifier guidance to come to our method? One would be that the classifier guidance will take the same data and then use a classifier's gradient to walk, or MCMC walk. So here, instead of that, you're taking the noise version of the data and then use the classifier guidance to walk. But then if you use a classifier guidance to walk on the noise data, you will end up at a noise state. So you still have to go back to the discrete space. And so here comes our gradient guidance.
In classifier guidance during the generative sample, it is applied in the noisy sample, right?
So if you use a classifier, like if you just use a classifier guidance in the diffusion model, wouldn't you use the same data samples?
Yeah, it's a noisy sample, right?
The classifier is itself trained on the data.
Yeah, classifier itself trained on noisy version of the data. The classifier, that, for example, in the forward process, you'll generate multiple noisy samples, right?
Yeah.
And the classifier is actually trained on these noisy samples.
I see. Okay.
Yeah, and in the backward, also they were used to give a proper guidance to the generative sample.
I see. Yes. So assuming that holds, you still use the classifier guidance to reach a certain region, which is going to be in the continuous region, but you still have to have this jump to the discrete space.
Yes.
And then you have to use that gradient guidance jump, because the normal jump is going to take you to, well, it's just not compatible with the gradient guided generation.
So your gradient guided you are using for the gem process, for the gem step.
We are using gradients for both steps actually. So the gradient guided walk would be almost same as the classified guidance, and then you still have to jump back to the discrete space using the gradient. So in our paper, we also show that, without the gradient and with gradient will make a difference.
Yeah, understood. But anyway, you are using the gradient of the noisy sample, right? Gradient with respect to the class label of the noisy sample, right?
Yes.
Why we have to give different name? Why we can't go with the literature used name like classifier guidance?
Okay, so that would be okay. So we took this model based on the walk jump sampling. So what they show is like in the walk part, you still have the walk part is not noise scheduled. So you don't have any noise schedule for walk. And so you just simply use some MC there. So the classifier is more common with the diffusion literature, and we have this method based on the walk jump sampling literature. So we try to stick to that. But we do have a lengthy discussion on classifier guidance in our paper too. So
you have explained in the paper.
Yeah, to not confuse anybody.
Yes. Okay, thank you.
Yeah. Any other question?
Okay. Good. I like to okay, thank you so much, Zarif. So I like to discuss a few things with you. So our iClear submissions didn't get a